Today, we're going to talk about something that is, frankly, beautiful. We're diving into the world of immunofluorescence, which is basically a technique that lets scientists paint a cell with light. It sounds cool because it is. This helps them see what's really going on deep inside, and that can unlock clues to pretty much everything. So let's get right into it. So let's start with a big question. How do you take a picture of something you literally can't see? I mean, cells and all the tiny proteins inside them are completely invisible to us. So how in the world do we get a clear image? How do we figure out where specific things are and, what they're up to? Well, the answer is a brilliant process called immunofluorescence. The best way to think about it is like a super specialized, high-tech biological photo shoot. We're using fluorescent tags to light up very specific targets inside a cell, turning this mysterious microscopic world into a vibrant, incredibly detailed picture. Now, every great photo shoot needs some prep work, right? And it's exactly the same at the cellular level. This first phase is all about getting our subject, the cell, ready for its big close-up. We've got to prepare the canvas before we can even think about painting. It all starts with 4 key moves. First, we grow the cells and give them a good wash. Then we fix them and permeabilize them. And don't worry, we'll break down exactly what those words mean in a second. And finally, you'll see this is a bit of a theme. We wash them all over again. So what is fixation? Okay, imagine you're trying to take a perfectly clear photo of a crazy, bustling city, but you're in a moving helicopter. It would be a blurry mess. Fixation basically hits the pause button on everything. We use a chemical, paraformaldehyde, to freeze the cell in that exact moment, locking all its internal machinery right where it is. Okay, so the cell is frozen in time, which is great, but its outer wall is still totally sealed off. To get our staining tools inside, we need to, well, open a few doors. And that's where permeabilization comes in. We use a gentle detergent called Triton X100 to poke tiny little holes in the cell's membrane. These become the gateways for all our dyes and antibodies to get inside. Now, here's something that is absolutely critical to this whole process: the constant washing. After pretty much every single major step, the cells get washed with a solution called PBS. This is non-negotiable. It's like a painter rinsing their brush before dipping it in a new color. It gets rid of any leftover chemicals from the last step and makes sure the next one starts on a perfectly clean slate. All right, with our canvas all prepped and clean, it's time for the really targeted part of the mission. We're not just looking at the cell as a whole, we are hunting for very specific proteins hiding inside it. This phase has its own precise sequence of events. First, we block. Then, we add our primary antibody. We let it sit for a while to find its target, that's incubation, and then, you guessed it, we wash again to clear out any of the antibodies that didn't find their mark. Before we send in our highly specific little seekers, we have to make sure they don't just stick to random stuff. The inside of a cell could be kind of sticky, lots of surfaces where things can cling. So we use a blocking solution, in this case BSA, to coat all of those random sticky spots. This ensures our antibody only binds to its one true target and ignores everything else. And here are our seekers. These are the primary antibodies. For this particular experiment, we're going after proteins that are super important for bone formation: ALP, OPN, and OCN. You can think of each antibody as a tiny, perfectly shaped key that is designed to find and lock on to only one of these protein targets. And this shot just shows you how precise this science really is. This is from a real lab protocol, and you can see those handwritten notes. Those fractions, 1 to 50, 1 to 100, they're basically recipes for diluting the antibody. Getting that concentration exactly right can be the difference between a blurry, useless blob and a sharp, breathtaking image. It's that critical. All right, this is probably my favorite part. The canvas is prepped, our targets have been marked by the seekers. Now, it's finally time to turn on the lights. So, remember, the primary antibody was the seeker. It found our protein. Now, we send in the secondary antibody. Think of this one as the illuminator. It's designed to find and latch onto the primary antibody, but it's carrying a special passenger, a fluorescent molecule called a fluorophore. For this protocol, it's Alexafluor 594. This is literally the light bulb that will make our target glow. But why stop at just one color? To really get the full picture, we need context. We need landmarks. So at the same time, we're also staining other essential parts of the cell. We use a stain called Phalloidin to light up the cell's internal skeleton, the cytoskeleton, in a bright green. And then we use another one called DAPI to stain the nucleus, the cell's command center, in blue. So what we're left with is this amazing color coding system. It's simple, but it's powerful. When we finally look at the image, we'll know instantly that anything glowing red is one of our target proteins. Anything green is the structure of the cell, and that blue area, that's the nucleus. We're creating a multicolored map of this tiny world. We are on the home stretch. All the staining is done, all the washing is done. Now we just need to prepare our cellular masterpiece for its grand debut under the microscope. The final 2 steps are pretty straightforward, but they're also delicate. First, we carefully mount the tiny cover slip that holds our stained cells onto a glass slide. And then, at long last, it's time to take it to the fluorescent microscope and capture the image we've worked so hard to create. But there is one final, crucial warning. As this note right from the protocol says, we have to protect the sample from light at all costs. Why? Because these fluorescent dyes are a bit like old color photographs. If you expose them to too much bright light, they actually fade away. It's a process called photo-bleaching, and it is the arch nemesis of a good fluorescent image. And after all that meticulous, careful work, this is the payoff. A stunning composite image where the red, green, and blue signals all combined. And it is so much more than just a pretty picture. It's a data-rich map that tells scientists exactly where their proteins of interest are living and working within the incredibly complex landscape of a single cell. It really makes you think, doesn't it? A technique like this gives us a window into a universe that exists inside every single living thing. Now that we have the tools to literally paint these invisible worlds with light, the only real limit is our imagination and what we choose to explore next.